you. Thank you so much for the kind introduction. I'm Kennedy from Purdue University, and sorry, how do I? You gotta go back. Okay, thank you. So today I want to talk to you about Schistosomiasis, which is one of the tropical neglected diseases affecting over 200 million people worldwide, especially in Africa. So this project focuses on the biology of Schistosomiasis. We are looking at the migration process, and it's a multidisciplinary work involving our colleagues in Kenya, collaborating with us in the US, and the specific topic that we are interested in is how the trematodes are able to move from the snails into the humans and transit through different tissues and back to the snails and continue the complex life cycle that they go through. So this is just a highlight of what happens actually during the infection process itself. I'm looking at the migration of the eggs through the host tissues, and you're going to look at, we start with the eggs laid in the intestinal tissues. You will see them penetrating the endothelium, going through to make the granuloma, and then they are released, they get entrapped, and some get entrapped, some get penetrated through, they get released, and all they go to the feces, and they go back to the snails. So this is a very complex process, and we still don't understand how the eggs, without legs and hands, how they actually transit through the tissues and back to complete the process. So we have a hypothesis that we've been pursuing in this work. We think that the immune cells that get recruited around the eggs, including the fibroblasts. So the host cells actually play a role here in trying to create forces that move the eggs. And we look at how the stress around the tissues are going to change during the recruitment process and the forces that are generated through that in collaboration with the host tissues. So the work that we are trying to do here, we have the in vitro work where we are developing some the microphysiological systems, and we are trying to build this sort of a chip here where we have the capillary, the stroma, the lumen, and right now we are trying to look at the migration process specifically in the stroma itself. And also we are doing another part of the work is being done in Kenya where we have in vivo studies. They are infecting animals and looking at the tissues, how the granuloma forms actually in vivo. So today, what I would like to tell you about is the work focused on mimicking the physiological environment using the hydrogels. We are doing the stiffness testing here. We want to develop something that mimics what happens in vivo because the metric stiffness is very important for the migration itself. So we are able to characterize the hydrogels and get the stiffness required. And again, we are using the PIV, which is a particle imaging velocimetry, to try to track the movement of cells within the gel that when they interact with the eggs. So we are having the bead assay here. As you can see, we have the fluorescent beads. We can track them using the PIV to generate vectors, and from the vectors, we can now tabulate the forces that they generate during the migration process itself. So for more information, please come to my poster. Thank you very much.